So let's talk about the genetic code, or the way that that messenger RNA is actually interpreted into an amino acid sequence. So first of all, every 3 bases code for a specific amino acid. There are 20 amino acids, but 64 codons. So codons, just the name for the 3-letter code. So if we have 20 amino acids, but 64 different codons, that means that there are multiple sequences that code for the same codon. In other words, an amino acid has more than one code. Now the codons for a single amino acid, let's say it is serine, I just picked one, are all going to be very similar. In fact, in many cases, the only difference in the sequences for a single amino acid such as serine is that the 3rd base differs. This is called wobble. Basically, this means that the 3rd base in the codon can change and the other 2 remain the same while still coding for the exact same amino acid. So one of the characteristics of our genetic code is that it contains wobble, or has the characteristic of wobble, meaning that that 3rd base can change without changing the amino acids. So it's really the first 2 bases in that triplet code that are important in determining what amino acid is being coded for. A second characteristic of our genetic code is something called degeneracy. So this is what I was talking about just a second ago, where more than one codon specifies the same amino acid. Now, I actually learned this as redundancy, but your book calls it degeneracy. So I try to use degeneracy, but occasionally I'll say redundancy. So the importance of wobble and degeneracy is that it limits the effect of mutations. So you can have a mutation in what would be the 3rd base of a codon and still maintain the same amino acid. Likewise, you can have certain changes that change the code in other ways, sort of, depending on the amino acid, and still specify the same amino acid. Another way to limit mutation is the fact that similar amino acids have similar codes. So what I mean by this is that amino acids each have specific properties, and we'll talk about those properties in a minute. And similar amino acids have similar properties. So if you change a sequence from one amino acid to another that has the same characteristics, you're likely not going to alter protein function very much, which means you'll probably still have a functional protein of some sort. Remember that structure is related to function. So if you change an amino acid due to a mutation, you're likely to change the function of that protein as well. So between wobble and degeneracy and the similarity of some amino acids, you can sort of limit the effects of mutation in a cell. The bases of our mRNA are read in groups of 3. Those are our codons. Codons are non-overlapping and have a particular reading frame. A reading frame is basically the 3 base pair lineup of codons. So in your sequence, you have a start codon, AUG, remember that codes for methionine. And wherever you start, you're going to set the reading frame. So after that, every 3 base pairs is going to code for a single amino acid. So let's say that this sequence here was preceded by AUG. and then our next amino acid without any skipped bases in the sequence and not overlapping with any of the part of the methionine codon, we get our next sequence of bases, or our first reading frame. Now, if this AUG is positioned somewhere else, we could actually get a different set of reading frames. So you can see if we start at the A, we have one reading frame, arginine, serine, and so on. If we start at the second base in this sequence, we get a different sequence because we are reading a different set of triplets, glycine, proline, etc. The third reading frame, if we start at the third letter instead, we get valine, etc. So essentially, there are multiple potential reading frames in any region of DNA because we have 3 at any one place in the DNA. And then also on the opposite strand, we have 3 more because remember, we can code on either side. So that reading frame is really important in setting what amino acid sequence you are going to have. Now, a couple of times we have talked about a frame shift mutation. A frame shift mutation is one that shifts the reading frame. Now, this is important because it changes all of the amino acids in the protein. And this is very likely going to affect the protein to an extent of it being non-functional. So frame shift mutations can be detrimental to a protein. Just as a quick review, we talked about some others. We talked about nonsense way back in chapter 2. That's where you introduce a stop codon. We talked about missense, which is where you change the amino acid. And then we talked about silent, which is where you don't change the amino acid. So this would be one of those wobble changes where you change the third base pair, and so you don't code for a different amino acid. All of these are likely to be point mutations, just one base pair, although they could potentially be more. Frame shift would either be 1 or 2 base pairs. If you insert 3 base pairs, you are inserting an amino acid, but you're not shifting the reading frame. It would be like just putting another amino acid here. We haven't changed the sequence, we've just added 1 in the middle. So here is our genetic code table. We've done this before, so this should look familiar to you. If you are reading your messenger RNA, you start with your methionine, A, U, G. So that is the amino acid that we code. Let's say we were coding with CCG. We would find C, C, G. That's coding for proline. We see degeneracy in all of these except for methionine and tryptophan. Each of those are only coded by 1 sequence. Most of them are coded by 3 or 4. You can see in, for example, serine, there are 4, proline 4, etc. Some are only coded by 2 sequences. You don't have to memorize those. You just need to be able to use this chart if asked. And actually, serine has a couple more over here. So leucine and serine also or together have 6 each. So there's a good example of our degeneracy. Remember, that is where multiple codes or multiple codons code the same amino acid. So 6 different codes encode leucine. We can see wobble with alanine, sort of, that little box is in the way. Any of these really. GC is present, the first 3, but any of the 4 bases in the 4th position code for alanine. We have 3 stop codons. Two of them are wobble related to each other. And then, of course, we have our one start codon, which in this case is labeled as methionine because we insert a methionine at that spot. That's our first amino acid. Now, if methionine is coded in the middle, it's just methionine. It's not another start codon. I got a little ahead of myself. So this is just another summary. AUG is the only methionine codon. It's also the start codon. And then, of course, we've got our other stop codons. Tryptophan, which is wobble related to one of our stop codons, is the least used amino acid. Also, interestingly, this amino acid here, isoleucine, is the least used isoleucine codon. Not really sure why. Possibly some advantages to not being able to wobble into a stop, or I'm sorry, not wobble into a stop codon. Possibly related to the advantage of having multiple stop codons instead of having another tryptophan codon. I'm not really sure about the isoleucine. Some of this is really just sort of products of evolution, like why is leucine coded by 6 and others are only coded by 2? There's not a lot of answers for some of those. But again, just make sure you guys understand how to use this in case you are asked to translate anything again. For this unit, I mostly want you to understand the idea of the codon and the wobble and the degeneracy. We're not going to be doing as much translating. So a few minutes ago, I referenced amino acids and said that amino acids have specific properties. There are 3 major groups of amino acids: those that are polar and charged, which means they are going to interact well with water. These are going to be your hydrophilic amino acids. So a protein that is high in hydrophilic amino acids is going to interact well with water, or maybe a region of a protein is high in polar charged molecules, and so it interacts well with water. We've got our polar but not charged group. That's our purple group here. And then we have our hydrophobic and nonpolar. So these do not play well with water. And so again, you can have a largely hydrophobic protein or simply hydrophobic regions of a protein. And then we have a few special cases. These guys don't really fall into any of those categories, have sort of their own characteristics. But each of these is going to give a protein a specific characteristic, or regions with characteristics. Now, if you were, say, to make a change that substituted lysine for arginine, you probably aren't going to affect the overall function of that protein very much. Doesn't mean you won't affect it at all, but if you substitute a polar and charged amino acid for another polar and charged amino acid, especially one that's got a really similar shape, you probably aren't going to affect things very much. However, if you substitute a non-polar hydrophobic amino acid for a polar one, you are probably going to change the function of the protein. So this is where we start to see the effects of mutation. When you change the amino acid, you change the properties of those proteins. And that's important because one of the things that happens after translation is that our polypeptide chains are folded into functional proteins. And if you have different amino acids, you get different folding and therefore different function. So this chart just shows you essentially where the amino acids are from one another. And you can see that if you look, the sequences are actually pretty similar. Isoleucine and leucine only differ by one base pair. It's not a wobble difference, but it is a first base pair difference. So again, if you were to mutate from isoleucine to leucine, you might not affect things too much. So our genetic code is universal, and this is important for a few reasons. So a universal genetic code is a code that, or it is considered universal. The genetic code is considered universal because all organisms across all kingdoms, that means bacteria, archaea, and eukaryotes, all use the same genetic code, and they use the same process to convert nucleic acid sequences to amino acid sequences. There are a few weird exceptions where variants of some of those amino acids are used, but in general, we can say that this 4 base pair code with the triplet codons, even what amino acids are encoded by what sequences, is conserved across all of our kingdoms. Now, this suggests that all living organisms can be traced back to a common ancestor that used this code. This makes a single common ancestor that is ancestral to all living species on this earth. This also shows the relatedness. So the genetic code and the fact that each organism is essentially a few steps in the genetic code away from another helps to explain how all these organisms are related. Now, organisms with similar genetic codes or similar DNA are going to be closely related because they have a common ancestor that shared that genetic code. Now, this is important for us. Let me back up before I say that. If a gene is produced by the same process, no matter what organism it is, that means that that genetic information can be transcribed in another organism and translated in another organism. So when we talked about horizontal gene transfer in chapter 11, horizontal gene transfer works, and horizontal gene transfer allows for that genetic material to be expressed in another bacteria because all organisms use the same code. So it doesn't matter where the DNA came from, it can be expressed and turned into proteins. This is also important for viral infection. Viruses commandeer the machinery in living cells to produce their own proteins and RNA in order to replicate as part of their life cycle. So their entire life cycle depends on this universal genetic code that no matter what cell they infect, they can commandeer that cell and use its machinery for its own purposes. And then, of course, selfishly, this is important for humans because we use this all the time in our labs. We did not do pGlow this semester. We did the CRISPR transformation this semester. Depending on what semester you're watching this, you may have done one or the other. But that genetic transfer of information works in other organisms. We can use gene transfer for biotechnological purposes because genes from any organism can be expressed in any others. So as just sort of a quick wrap up for this chapter, we talked a lot about regulation of transcription. We talked a tiny bit about regulation of translation. Proteins are or protein function can be regulated by the modifications that are made to the protein. Now, we are not going to talk about how proteins are actually folded, how they are processed, except to say that there are specific modifications that are made to proteins to make them functional in the end. And so this can lead to some types of regulations. This image here just shows some of the ways that proteins are modified. So for example, tyrosine, serine, threonine, histidine, and aspartic acid are phosphorylated. That means a phosphate group is added to them. And those modifications are going to further affect the shape and function of a protein. And it's going to affect how it interacts with other proteins and how it does its job in a cell. Each of these modifications are covalent modifications. That means it's a covalent bond, which means it's sort of a permanent modification to the protein. So once all the folding and post-translational modifications occur, you have a final functional protein. So just as a quick review, we started in chapter 12 with transcription. Then we moved on to translation. And although we didn't talk about it, after translation, we get to that functional protein product or functional gene product. So this example here is hemoglobin. Hemoglobin is the molecule in our blood cells that carries oxygen. And this little heme group in the middle is the molecular group that actually binds oxygen to it. Hemoglobin is made up of 2 alpha chains and 2 beta chains that are each encoded by their own gene, each folded and put together before this polypeptide can be functional. And then of course, once we get the functional product, we get our fully functional hemoglobin gene. And that is built into our red blood cells. If you remember from our discussion on sickle cell earlier in the semester, a mutation in the beta chain of the hemoglobin is what causes that sickle cell shape. The hemoglobin changes shape because there's a mutation in that beta chain gene, and that causes the overall structure of the red blood cell to take on that sickle shape. So there again, we have this link between the structure of that beta chain and the function of the beta chain. And a good example of how a mutation can change the structure of a particular protein.